Sorry I couldn't be here today with you guys to go through this intervention. I'm hoping that this way will still be beneficial to you and get you ready to be able to take the C5 mastery check tomorrow. So we're just going to start going through this fairly quickly. However, I'll try and explain things as we're going through. So, number one, we're going to start with our flow chart on cell transport and try and get you guys to understand kind of what's happening. There's lots of different scenarios and things like that, and that's really the hardest part of this, is being able to understand what it means when we're talking about active and passive transport and all of the different kinds. Okay, so starting out, let's go with right here. We have cell transport, and it can go two different ways. Now, number one, you can either have passive transport or you can have active transport. Now, the reason that it would be called passive transport is because we would have something called a concentration gradient. Okay? Now, a concentration gradient is where you have a really, really high concentration in one area and a really low concentration in the other area. So, what that would look like is if we had something of where we have a whole bunch of little tiny dots over here, okay, and then over here they'd be much more spread out. That is what a concentration gradient is. And so then what would end up happening in that situation is that the stuff right here that we would call a high concentration would move towards a low concentration. Okay, so that is what we're talking about when we're talking about passive transport. Passive transport always has to do with a concentration gradient, and that concentration gradient is going to move from high to low. So on the other hand, if we don't have a concentration gradient, or if something is just way too big to be able to fit into through the cell membrane, we end up having to use something called active transport. And whenever we have active transport, it just can't go naturally like it does with passive transport. So what ends up happening is that you have to use energy, typically in the form of ATP, to be able to move those other types of things across the cell membrane. So, again, remember, anytime that you're doing active transport, energy is required. And you would not have the concentration gradient to make it go naturally. So the different types of passive transport that we have are simple diffusion, facilitated diffusion, and osmosis. So when we're talking about simple diffusion, what that means is that we are going to be small and non-polar. Okay? So really only non-polar things can be able to move across the cell membrane without any help. So really the only things that we've ever even talked about would be oxygen and carbon dioxide. Those two molecules are pretty much the main ones that can just diffuse across the membrane without any help at all. And so just remember that if it is simple diffusion or doesn't require any sort of help at all, it has to be small and non-polar. Anything else that is polar, if it tries to get through, it's going to hit the fatty acid tails of the phospholipid bilayer, and when that happens, it's going to boot them right back out, and it's just not going to work very well. So, the next part would be facilitated diffusion. And facilitated diffusion is going to be something that is small and polar. So, this would be anything that has a positive or a negative charge. Maybe you guys can hear my kids screaming in the background. Hope you enjoy that. But anything that is small and polar, has a positive or a negative charge, it's not going to be able to make it through that cell membrane by itself. It's just going to keep on getting kicked in and out of that cell membrane over and over and over again. So you have to have a special way for it to be able to get in and out. And so, like we've talked about before, you have to build like a doorway or a passageway. And the way that you do that is that you take a protein that has a shape with a hole in the middle of it, and then it can allow those things to go in and out. So anytime you have facilitated diffusion, even though there's a concentration gradient, you have to have that passageway or that doorway made out of protein to be able to have things to go back and forth between that. And then the last part that comes up is osmosis. And osmosis is going to have to do with water. Now, water gets its own special thing because even though it's a polar molecule, sometimes it can actually go through the membrane, and so it's kind of that in between. Plus, water is extremely important, and it's always moving across cell membranes, and so it just gets its own special name. So anytime you have the diffusion of water across the membrane, it's always going to be osmosis. Now, on the other side of things, going through all of the specific types of active transport, again, that require energy, there's going to be two main ways that active transport is going to be used. Number one is if something is just way too large. If something is way too large, it can't make it through even those normal proteins. So when we're talking about a protein, we're talking about something that is going to be like 300, 400 pieces of amino acids big. Whereas when we're talking about something like carbon dioxide, it's going to be smaller than just one of those pieces. So that is a huge size difference that you just don't, that just can't go in and out. And so you have to be able to make these vesicles, which, if you remember, are kind of the bubbles of phospholipid bilayer that can encapsulate something and then be able to move it into the cell or out of the cell. And so when we're talking about vesicles, we have two main different kinds of things. Now, if you go into the cell, that is going to be called endocytosis. So endocytosis always has the vesicle is going into the cell. So endo is going to mean into, cyto means cell. So it's going into the cell. Now, if we are on the other side, and we're talking about exocytosis, exocytosis is going to move out of the cell. And when something moves out of the cell, it would typically be something that you would just need somewhere else in your body, and it would be able to move there. So again, if we look at the word exo, means outside or out, and then cyto again means cell, so it means out of the cell. Now, we could get a little more complicated. I don't really want to get more complicated with you guys. And so there are different types of endocytosis. There's phagocytosis, pinocytosis, and receptor-mediated cytosis. But really, on a test or something, if you ever see cytosis, that basically is going to tell you that it is some sort of vesicle. And as long as you know that, that should be sufficient to be able to get you through any sort of question on the test. So, the only other type of active transport that there is the pump. Now, the pump is basically moving small molecules, however you don't have a concentration gradient. So, let's say that your body needs a certain amount of salt to survive within its cells. However, you're in a solution of where salt just keeps on getting pulled out more and more and more out of your cells. Well, if you're going to die, your body isn't going to really want that. And so what it's going to do is that it is going to start using energy to start pulling that against the gradient. Okay? So this would basically be like pushing something uphill. Now, if you're at the top of the hill and you let it goes naturally and it falls down to the bottom. That's passive transport. But in this kind of scenario, what's happening is you're at the bottom of the hill and you're trying to push it back up, and that requires a whole lot of energy to be able to push something up the hill. So, whenever you think of a pump, just think of something that you have to go against the gradient. And if you are going against something, that means that you are going to have to use energy to be able to get it there. Okay, so now that we've talked about this, you guys are just going to need to be able to study this and go through all of the different scenarios and stuff, and just start thinking about it and putting it together. If you don't put in the time to just kind of look at the different ways, it's really going to get confusing on a test. So if you need to, go back through and play this again. However, we are going to move on to the next part of this, which is talking about scenarios of where you would have osmosis, and diffusion. So, in our first thing that we're going to have is that while I'm doing this, you're going to see a couple of different things. So, number one, I'm going to have something like H and L. And H is going to mean high, and low, L is going to mean low. And then I'm going to put something right after these of where if you see W with it, it's going to mean that I'm talking about the concentration of water. And if you see S, that means that I'm going to be talking about the concentration of the solute. Okay? So solute could be anything like salt, carbon dioxide, oxygen, sugar. So whatever you want S to mean, I don't really care, but you need to understand that I'm talking about the concentration of the solute. So, in our first scenario, we're going to start out with high solute on the outside, and we are going to, which would mean that if it's high solute on the outside, that means that it's got to have lower water. Okay? And then inside of our cell, so this thing right here, this is our cell. We're going to have low solute because, again, if we have a high on the outside, higher on the outside, the opposite has to be true, that it is lower on the inside. And so then, if we have low water on the outside, that means we have to have a higher concentration of water on the inside. Okay, so this is how we talk about that word called tonicity. Okay? So with this one that we're looking at first, we'll just kind of come right in here. And we've got on the out in the solution, we have this, the high solute. If it is a high solute, that is the one you want to pay attention to label it either hyper or hypotonic. And since this one is high, that means that this is going to be hypertonic. Okay? So that means that this is always going to be hypertonic when you have the high solute. Now, we're not really going to worry about the solute right now. You guys should be able to figure that out. But the most important thing for you guys to understand in this scenario is to look at the water. So the water in this scenario, we have higher on the inside and lower on the outside. And since we're talking about osmosis, that is a type of passive transport, and so it's always going to go high to low. So water in this scenario is going to move high to low. Now, if you're moving water out of your cell, what's going to happen? It's going to shrink down and shrivel. So, in a hypertonic solution, a cell is always going to shrink or shrivel down. On the other hand, if we go over to this one, and we take this, and on this we go the opposite of where the concentration of your solute is lower on the outside, which would mean it would have to be higher on the inside. And then we could figure out the water as well, because that would mean that the water would have to be high on the outside, and the water on the inside would then have to be lower. Now, because this is lower on the outside for the solute, that is going to be what we call hypotonic. So, whenever you have the lower concentration of the solute on the outside, that means it's hypotonic. And because we have a type of passive transport again, it's going to go high to low. So in a hypotonic, what's going to happen is that we are going to have the water move in, which will end up making that start to swell. And it could even start to swell to the point of where it explodes. So, just to recap, when you're looking at these problems, it might give you one of the things, two of the things, but it's probably not going to give you all four of these. And so you're going to have to kind of fill in the blanks here. So number one, always look at the solution, okay? The solution that everything is sitting in. If it has high solute, that means it's going to be hypertonic. If it's low solute in the solution, it's always going to be hypotonic. And then just remember that things are just going to go opposite. So if you're higher on the outside, you're going to go lower on the inside, and then water is going to be the opposite of the solute. So you're going to have low water and high water. Okay, now that we've finished tonicity, just something that has to do with tonicity, and I didn't teach you guys this, so I'm going to go through it because it is going to show up on the retake that you'll be taking. So, yeah, let's just hop over to that, and that is going to be this. Now, when we're talking about an animal cell. An animal cell is going to follow the same old rules that we were talking about. If it's in a hypertonic solution, an animal cell or a bacteria cell or a fungal cell, they're all going to shrink. Whereas in a hypotonic, they are going to expand over and over again, and they could potentially burst. Now, isotonic for an animal cell, bacterial cell, that is what they like to have. That helps to keep them normalized and doing well. On plants, though, it's a little bit different. So when we're talking about plant cells, if you're hypertonic, the same thing's going to happen, because with a plant cell, what you have to remember is that there is this thing right here, okay? The cell wall. And it is like a big, heavy protective jacket. It's really not going to change shape that much. It can't change shape that much. And so inside of here where you see this green stuff, that is going to be the cell membrane of the plant cell. So in a hypertonic, just like an animal, what's going to happen is that cell membrane is going to shrivel and shrink. And so that's what you're seeing here, is that in a hypertonic, the same thing's going to happen. It's going to shrivel and shrink. And that's what that word, the plasmolized, there we go, plasmolized means. Okay, now, with an isotonic in a plant cell, that is usually what we call when its vacuole isn't going to be the fullest that it can be. And so, even though it still has sufficient water to be able to survive and things like that, it's not going to be as healthy as it could be. So, whenever you see isotonic in a plant, that just means that it would be wilted. So, when it's isotonic, it's going to be wilted. Which means with a hypotonic, you fill up that vacuole extra full, and then all of that pressure will push out. Now, in your cells, if that much pressure was in there, they would just explode. Okay? However, with theirs, what ends up happening, it pushes out, but it doesn't explode. So the normal everyday plant that you see is going to be in a hypotonic solution. That's why the soil always has to be damp for your plants to be as healthy as they can be. So, just a quick little review on that is this one, you're pretty much dead. On an isotonic, you'd be wilted. And then on a hypotonic, then you would just, this would be a normal, healthy, rigid, plump, firm plant. Okay? So, I don't think you really need to memorize those names, but just kind of understand what would happen in a hypertonic, hypotonic, isotonic for plants. Alright? And then, really, that is the majority of everything that you need to be able to know for this test. And you might need to practice some of these, like osmosis diffusion. Actually, let's go back there. That's probably a good thing for us to do real fast. Is with these, you're going to see on some of the problems, you're going to see this, and then you're going to see a line down below, and it's going to say whether it's osmosis or diffusion. So it's always going to be osmosis, so always put osmosis no matter what. Unless it's isotonic, then you don't have to. But anytime you have a concentration gradient, you've always got to put osmosis. Now, the only other time that you would put diffusion is if you have one of our two nonpolar molecules. So O2 or CO2. Those would be the only instances where you would put diffusion. Okay, so anything else, don't bother putting anything other than osmosis. Alright, guys. I hope that answered some of your questions. I'll be back tomorrow to help with anything else. Thanks. Bye.